Yep.
Does the quiz open till midnight?
11:30
Tonight, right?
tonight.
Okay.
All right. Up a little loud. See here. All right, we are live. We are streaming. All right, so just a couple of housekeeping. Quiz due today. I think I have it set to close at 11:30 tonight. So if you haven't done it, you better get to it. Make sure you review those intro lectures. So what is ecology and the methods in ecology. Make sure you review those. That's what those quiz covers. All right. Lab this week again. Wednesday, Thursday labs. Collect some data. Next week, we are learning about this Hardy-Weinberg equation. Learn how to use the Hardy-Weinberg equation. So bring your calculator. You will need a calculator. Scientific calculator is preferred, but regular calculator will work. All right. Any questions? No. All right, let's continue this. All right, Calvin's here. Cool. All right, so we left off with Hardy-Weinberg equations. You know, key point for Hardy-Weinberg is that if the assumptions are true, if all of these four assumptions are true, then our population is not evolving. All right. So if the assumptions are true, then we say that it's in Hardy-Weinberg equilibrium. there's no evolution. When we do have evolution, at least one of these assumptions is violated. All right, do we know which one? No, we don't really know which one, but we can kind of investigate it. That's kind of why we would do science, is we see, okay, this population is evolving. Let's figure out why it is. Let's figure out what's going on. So what we're going to do is step through these assumptions, because violation of each of these assumptions leads to a mechanism of evolution that, if you've had the evolution course, you've talked about. All right, so the first one was genetic drift. This is if we violate assumption number one, meaning we're working with a small population size. Genetic drift is the stochastic shifts in our allele frequencies. This is the random change. It's just random chance happens. All right, we said it's most important in small populations, but it's not the population size that really matters. It's how many of those individuals are actually taking part in random mating. And that population is the effective population size. So your population could be up here, population size could be up here, but your effective population size is down here. We need that effective population size to inform us about the effective drift, or potentially how much drift will influence our population. So key point here is as this difference between our population size and the effective population size increases, the effective drift will increase. Because normally what you see is you have a large population size to begin with. But as our effective population size starts to get smaller and smaller and smaller, you increase that difference, and now you get the effective drift. So these graphs represent an example of it. So I have handouts for the presentations. The handouts have some of these graphs, some of the figures. If you wanted to download them and work with them so that you have them and you don't have to draw them, that works. You can do that. All right, so here's an example, 4 different graphs where I change the effective population size just so you can see what happens. All right, so in each of them, what we are doing is looking at the frequency of the A allele, and I start at 50%. So 50% of all of the alleles are this big A allele. And then I'd go through a period of random mating, all right, no selection, no mutation, no migration, none of that. All right, and just run it out for 100 generations. And what do we see? Well, if we start at a large effective population size, N1000, all right, you get fluctuations. You get random fluctuations. And you get this line that is very jagged. And for the most part, it moves around this 50% point. It mostly stays where we're at. If we take 1/10 of that population size, we start to see much larger fluctuations. And not only that, but out of our 5 runs, I have 1 run where that allele was completely lost from our population, and it happened around generation 60. If I go half of that population size, you see even more fluctuations. And now, out of those 5, I have 3 populations, 3 simulations where our allele was fixed in the population, where we hit to 100%. And those happened later on, but 3 of them all happened around generation 90. And then in our small effective population size, where we start with 10 individuals, and we keep it at 10 individuals, all 5 simulations ended up with either fixing of that allele or loss of that allele. And when they happened, all of them happened in under 50 generations. All right, so as our population size gets smaller, we start to introduce chance events that can wreak havoc in our population. And how does it do that? It basically is going to randomly eliminate an allele. All right, in this case, 4 out of 5 times we fixed the allele, so we lost the recessive allele. 1 time we lost our dominant allele. So generally speaking, drift is going to cause a decrease in our genetic diversity. It's always going to happen. It's going to happen at some point. And that means this loss of diversity is that we're going to fix one of those alleles. Do we know which allele? No, it's random. But even in this large population size, eventually one of those alleles will be lost from our population. It might take 10,000 generations. It may take 1,000,000 generations. But the role of drift here not only is to introduce randomness, but it causes a loss of genetic diversity. All right? So if we have a small population size, we introduce randomness into our populations, and the consequence of it is a loss of diversity. Questions on drift? All right, second one is gene flow. This is violation of assumption number 2. Violation of assumption number 2. Gene flow is the gain or loss of alleles due to movement. So you have individuals that move from our population. They either move into it or they move out of our population. Those individuals are going to bring their own genome to our population. And as long as they have the same traits, same loci, all right, we can introduce new alleles. Now, gene flow isn't just the migration of individuals into or out of our population. In order for gene flow to occur, that movement must be disproportional. It must be non-random, which means we have to have a net exit or a net entrance of our alleles. And it's a specific allele. So if we start with our population 1 with, let's say, 50% big A and 50% little a, all right, and we have migration and we lose, let's say, 25% of the big A. If we have 25% of big A coming back in, that's not gene flow. Basically, what we've lost is what we've gained. So our frequencies for this population, even though we had migration, the frequencies stayed the same from generation to generation. But if we have 50% loss or 25% of loss of that dominant allele, but only 10% come back, now we have disproportional flow. And what we've done is changed our allele frequencies, and that change is directly due to migration, and we call it gene flow. All right? Questions? Now, where does gene flow come in? Gene flow's effect is that it can either oppose the forces of selection or it can reinforce the forces of natural selection. All right, so let's say allele A, our recessive allele, is naturally being selected against in our population. We've got selection. This is not a very good trait for our environment. Selection's trying to remove it. All right, selection is going to do a good job of getting it really low in frequency. But if we have gene flow, we can have individuals with this recessive allele bringing it back into the population. So instead of selection almost taking it out, we have gene flow trying to recover it. It also works the other way. So let's say gene flow or selection is trying to remove that allele. If those individuals sense that they're not doing well in the environment and they move, now we've just magnified the overall effect. We are more rapidly losing that recessive allele. I'm going to note that this is important in isolated populations. So in populations that are isolated and not near any other populations of its species, you're not going to have this gene flow. And if you're not going to have this gene flow, then when selection acts, it's not going to be opposed or reinforced by gene flow. Gene flow is not occurring. So in these isolated populations, then, natural selection can become very effective at altering our population. If we're in a population where there's others around and our individuals can move, can freely move, then selection might not be as effective at generating good traits. All right, hopefully you got all this. All right, third is mutation pressure. Mutation pressure is when we violate assumption 3. Assumption 3, no mutation, right? We said mutation, increases our genetic variation. It's like the ultimate source. All of our variation came from mutation, all right? And then once we have that genetic variability, then recombination can come through and start making new combinations. So what's mutation pressure? Mutation pressure is defined as the changes in the allele frequency due to the origin of new alleles in our population. All right? And I think we already gave a definition for mutation, which is the random change in the DNA sequence of a gene. Now, mutations are rare. All right? Mutations are rare. And because they're rare, they tend to have a very small effect on the overall allele frequencies. And we usually ignore their impact on the populations. But that's not to say that they actually don't play a role. Their role is to produce new alleles on which drift and gene flow and selection can act. It's just unfortunate because when mutations do occur, you normally have that one allele that appears because of mutation. And there's a decent chance that drift is just going to take it out. That individual has to reproduce. Not only do they have to reproduce, but through meiosis, whichever gamete that they give, that gamete has to have that allele, that new allele. So it's kind of hard to get it to persist, but they can. I mean, it's there. When you start dealing with large enough numbers, all right, large enough population size, it will eventually happen. All right? Yeah, it's very unlikely that I'm going to win the lottery. But if I play it for the next million years, every day, I'm bound to win it. If I don't, then I'm just extremely unlucky, and I shouldn't be here. But that's the same idea with mutation. All right? So genetic drift removes genetic variation. Mutation increases genetic variation. Leave that up. And you can see that I could really just kind of fly through these things, and then you don't get the notes, you don't get the slides. All right? You good? All right. Now, assumption number 4. Assumption number 4 was no differential mortality or reproduction. All right? So differential mortality basically means that some individuals have a higher chance of dying than other individuals. Differential reproduction means that some individuals are more likely to mate than other individuals. All right? We said that differential reproduction is basically non-random mating. So if we don't have differential reproduction, then we have that random mating. But we also have the possibility of sexual selection happening. Individuals choosing their mate, and they're going to choose their mate based on certain traits. All right? Those traits not only had to find a mate and try to win over a mate, but they had to survive throughout their life just to get to that point. All right? And yeah, it's random chance of dying, that happens. All right? But we say differential mortality can't exist because each individual has to have the same probability of getting to that point to reproduce. If we violate those assumptions, then what we have seen now is natural selection working. All right? So natural selection is going to occur anytime, whenever we have differential survival and or reproduction, meaning that one genotype is going to do better than another. And they can do better than the other at either surviving or finding a mate. Doesn't matter. Or perhaps both. All right, so this is our natural selection. and I'm sure you've probably already heard or seen natural selection. We quantify the effect of natural selection by using something called this selection coefficient, S. So as you'll learn in lab next week, this selection coefficient, S, can be incorporated into our Hardy-Weinberg equations to make predictions about the effect of selection on our allele and genotype frequencies. All right, so what is this selection coefficient? This coefficient tells us the proportion of a genotype that is not represented in the next generation due to death or reproductive failure. All right, so it's a proportion of a genotype that is not represented in the next generation due to death or reproductive failure. So I'll let you get that copied. And you're not getting cramps on you, are you? All right, so our selection is going to remove a proportion of our individuals from the population. All right? They're going to remove a proportion of a specific genotype, depending on what we're selecting against. All right? And we're going to model that proportion that gets removed using this selection coefficient. So our example here, and I believe this diagram's in our book, if you have the book. This diagram, all of these diagrams, well, most of these diagrams are also posted. At least these nicely generated diagrams are posted. All right? But this is what happens. We start with our population. All right? This is all the individuals there, and let's say selection acts on this population either during reproduction of our population or before or immediately right after our population. Selection happens between generation 1 and generation 2, and our selection coefficient is going to tell us what proportion of these homozygous recessive individuals, these little a individuals, get dropped from our population. And that means then, once we drop them, our second generation now has fewer of those individuals than what we would predict by chance alone. All right? And now I'm going to note that for this class, anytime we model selection, anytime we utilize this selection coefficient, we're always going to be selecting against that recessive allele. All right, so you're going to have to be homozygous recessive to express the trait. That's the trait that selection is going to take out. But in real life, selection can act on the dominant trait as well. All right, so if you're expressing the dominant phenotype, selection could take you out if you have a lower fitness. All right, but note, just for this class, we're going to emphasize this to say that when we model selection, we're always going to be selecting against that recessive phenotype. All right, so let's look at our relationships. S is a proportion. And just by mathematical rules, our proportions range from 0 to 1. All right, we can't go above, we can't go below. All right, we can't have 110% of our population get removed. That's just doesn't make any sense. It's when I hear, oh yeah, I gave 110%. You must have failed math. I'm sorry. Our proportions range from 0 to 1. All right, so if we have a selection coefficient of 0, we have no selection. 0% of our individuals get removed. They all are there to randomly reproduce and get to our next generation. If we have a selection coefficient of 1, it means we're working with a genotype that is 100% lethal or 100% sterile. They either can't survive to reproduce, or they survive to reproduce, but they just can't reproduce. They're sterile. All right, so those are our relationships. If it's 0, it's no selection. If it's 1, we're 100% lethal. So if we do have selection, then our selection coefficient can't be 0. It's going to be larger than 0. And the reality is that most of our selection coefficients are going to be less than 0.1. A lot of them are like 0.01. to like 0.03. But our range of selection coefficients and the value of our selection coefficients kind of depends on the function of the trait itself. So things that have more to do with like life history types of aspects, so survival, reproduction, they're going to be under stronger selection if there is selection. So their selection coefficient is going to be larger. Then you have a trait that might not really deal a lot with life history. Maybe it's about acquisition of resources, and while your trait allows you to find the resources to survive, it might not be the best option. Well, we can still select against it. It's just not going to be a very strong selection against it, so we'll have smaller values. Now, what's the effect of selection on our allele frequencies? Well, when we have selection, we're going to push an allele towards fixation. We're going to push it towards 1. And in this class, as I said, our selection is going to be against that recessive allele, so the effect on our populations is that our allele frequency P gets pushed to 1. We're going to make our population consist more of that dominant allele, that dominant trait. All right, so here's a graph that kind of shows that. Hand-drawn graph, I think it's pretty easy for you just to kind of transcribe it. So on this graph, what we are graphing is P, which is our allele frequency. It's the frequency of our dominant allele. And our frequency ranges from 0 to 1. The frequency is just a proportion, all right? 0% to 100%. And what we're going to graph it against is time. So time kind of represents our generation. So every generation we reproduce, all right? And with every generation, we're just going to add more and more time. All right? So what this graph is going to show is how our allele frequency changes for 3 different values of our selection coefficient. So we start with a selection coefficient of 0. That means our selection is not happening. No selection whatsoever. And if it's not happening, then our allele frequency should stay at 0.5. It's just a flat line, shouldn't change. Now we know in real life we have genetic drift that's going to kind of cause it to fluctuate, but in a theoretical population where there is no drift or gene flow or mutation, and now no selection, our allele frequency stays flat. As we increase selection pressure, so as our selection coefficient increases, we start driving our population towards fixation of that dominant allele. How quickly we get there depends on the coefficient itself. So if we could have a selection coefficient of 1, meaning the trait's 100% lethal, and compare it to a situation where our selection coefficient is 0.5, we get to fixation much, much faster if our trait's 100% lethal than if it's not. But we never really reach 1. Our allele frequencies never really reach 1. Selection can't ever get us there. Why can't it get us there? Well, it's because that trait, so we talk about dioecious individuals, and we talk about dominant and recessive alleles. We assume that our trait is expressing complete dominance. So the only way that you can express that recessive trait is to be homozygous recessive. All right? But eventually we get to a point where it's extremely unlikely that we would have 2 heterozygotes get together, reproduce, and produce that homozygous recessive individuals. So we get to a point where selection appears like it's stopped, even though it hasn't. But we don't see the change because all of those recessive alleles are now in our heterozygotes, and they're not being expressed. So we kind of get close to 1, but we never actually get to 1. Selection can't ever push us there because the recessive trait doesn't get expressed. And we have a term for that phenomena, and it's called heterozygote refuge. So it means that our recessive allele is hiding in the heterozygote, and what's it hiding from? It's hiding from the forces of selection. So if we want to try to remove this trait, selection can't do it all by itself. Selection will get us close, but then we need another mechanism of evolution to get us all the way. And that mechanism is either gene flow or drift. Mutation's unlikely, it's rare. All right? But we need gene flow or drift to happen. So when you have those handful of individuals that are heterozygotes still left in our population, we are relying on just random chance to say that some of those individuals don't reproduce, and thus their recessive alleles are lost from our future generation. Or we're relying on those individuals to say, hey, you know what, I'm fed up with this population, let me move, let me try to find a new area, new habitat that I can raise my kids and so forth, and they leave that population. That's what we need. So selection is also going to cause fixation of alleles, but it can't ever get us there all the way. We need some other force to actually get us to that 100%. Questions on that? All right, so selection can take on several different forms, and I'm sure you've seen this. So we'll just kind of very quickly breeze through this. So 3 types of selection that we've encountered, or that you've encountered, is directional selection, stabilizing selection, and disruptive selection. All 3 of them can occur. All right, so start with directional selection. Directional selection is a situation where one tail of our phenotype is favored. All right, so you've got to remember, we kind of introduced Hardy-Weinberg with a very simple case where it's just a single locus defines our trait. That locus has a dominant allele and a recessive allele. Simple, right? Very easy to conceptualize. But in reality, a lot of our traits are what we call quantitative traits. So there's multiple loci that control for them. And it's not just, do you have the trait or not, but our trait now takes a range of values. So this is what we're talking about, is we have selection on those range of values. So directional selection is a situation where one of our tails is favored. So here, our example is Darwin's finches down on the Galapagos. All right, so we know down there that based on the rainfall, you can alter what the dominant, the most abundant seeds are down on those islands. And those seeds are the food source for these finches. All right, and based on those seeds, it kind of, whatever seeds are available, there's always a best beak at crushing or cracking those seeds. So as you get, more larger hard-shelled seeds, all right, it's going to favor beaks that have more mechanical advantage at cracking those seeds, so larger beaks. As you get smaller seeds, now having that big, massive beak isn't really good at picking up the smaller things. All right, I don't know, you're probably not there yet. Some of you might be. When you have kids, I mean, it seems like they can pick up the small little beads and the small, sparkles very easy, and then I try to pick them up, my fat hands just can't get them. All right, same concept. All right, really tiny seeds. All right, the big beaks don't work, so we've got movement back. All right, so this is directional selection. One tail of our phenotype. So I should say, on these curves, what we are representing is beak size is the x-axis. So this is the range of phenotypes that can occur in our population itself. So we could have small beaks, all right, all the way down here. We can have large beaks. And then the y-axis tells us how many individuals have that beak size. So this is a histogram. All right, that's what it is. So you can see that most of the population, the high N, the large N, has a beak size that's kind of, let's say, intermediate. Smaller to intermediate. So if we have directional selection, what we say is that these larger beak sizes are favored. So this tail is favored. And then over time, what's going to happen is that these individuals reproduce more and leave more offspring, and their offspring have these larger beaks. And then over time, we see a shift in our histogram, in our phenotype curve, so that now most of our population is going to have the larger beak size. We still have a range of beak size. We still have smaller beaks and larger beaks, all right? But most of our population is going to have that individual. So directional selection is that we see a gradual increase in our trait or a gradual decrease in our trait. That's our directional selection. And you don't need this picture of all the finches. That was just an example to show you some of them. Did you guys get this graph? Good. All right, stabilizing selection. Same type of graph. All right, we start with the black line here. All right, so the black line is what we see out in our population. It's the range of phenotypes, and what we're depicting is birth weights. All right, so most mammals exhibit this type of selection. All right, so our birth rates could be small, it could be large. And these birth weights are going to be the weights at a full-term birth. All right. We have stabilizing selection that happens. And the stabilizing selection is favoring the intermediate portions of our curve. All right, these individuals do best. So over time, we have a compression of our phenotypes that produce population where most of our individuals have a small range of birth weights, and it's going to be more in the middle of our range. Now, most mammals exhibit this type of selection, the stabilizing selection. Most mammals exhibit this in terms of birth weights. Why is that? Why is that? Why wouldn't we go for larger individuals?
Because even though they might be stronger once they come out, it's not good for the mother and she might die or have trouble getting out.
Yeah, so right here, if we go for the larger, so you actually covered both of them. So birth weight is related to survival, all right? So you'd think that larger individuals are going to be more able to survive after they're born, which is true, all right? And that's why we don't favor the small individuals, that their survival after birth is much, much less. But we have a constraint, and that is as you get larger and larger and larger, it becomes harder and harder and harder for you to get out of the mother, to travel down the birth canal. All right, the birth canal is only so large. All right, and even though it can fit, it could be like right at the maximum end edge, which now places selection pressures on the mother, or risk of mortality increases for the mother itself. So you've got this sweet spot, you can say, where selection is favoring more of the intermediate traits, kind of a balance between survival after birth and actually being born, making it through the birth canal. Questions? All right, our last form of selection is disruptive selection. Disruptive selection is a situation where both tails of our phenotypic bell curve are favored. All right, so here we have our whole population. Same type of histogram. This is phenotypic curve. All right, so we've got the range of phenotypes that are exhibited by our population. So there's our top, here's our bottom. In disruptive selection, both of our tails are going to be favored, meaning we're selecting against that central part of our distribution. We're selecting against the intermediate trait. And then over time, what we start having is a separation of the traits so that we have a chunk of the population that has a smaller phenotype, right over here, and then you have a chunk of our population that has the larger phenotype up here. All right, so we get a split here. Now, what's the, yep?
Is that kind of like like creation of new species, or would it help with that, just like separating them from one part of the population commonly having one phenotype and the other having?
It could. It could. It could definitely lead to species separation or formation of a new species. But what is a new species? We'll leave that for Dr. Dollar. All right, we'll leave that for Dr. Dollar. All right, so we are separating population. Now, the thing is with disruptive selection, and why we don't really want to go to the separate species, is that we can still take these individuals, an individual of the large phenotype, individual of the small phenotype, reproduce them. they still produce viable offspring, and the offspring can still reproduce themselves. All right? They can still do that, so that they're still the same species. Our example, now, I should say, disruptive selection is pretty uncommon. All right? And it's actually somewhat hard to show. But we do have some examples of disruptive selection occurring. One of them is in the monkey flower, Mimulus luteus. All right, so the monkey flower, this is kind of an illustration of it, has a range. And a paper came out that looked at this type of selection. So the flowers themselves can be large or they can be small. All right, and when we look at flower size, there is evidence of directional selection selecting for larger flowers. Larger flowers attract their pollinators, which happen to be insects and birds. Insects and birds. So the larger flowers do better overall, directional selection. But you have this other aspect. And I know my class in lab last week talked about the desert willow and how you've got this landing strip for the pollinator. All right? It's called a nectar guide. All right? And the nectar guide is going to be colored. It's going to be emphasized to try to attract our pollinators. So the nectar guide, all right, is what's under directional selection. Not directional selection. It's under disruptive selection. So the nectar guide can be large or it can be small. And which one is favored depends on the pollinator. Now here's the interesting thing, is that the pollinators of this plant are hummingbirds or moths, other insects. And they're active at two different times. All right? So one's active during the day, one's active at night. Those flowers in an area that are predominantly pollinated by these insects exhibit, or they're favored, much larger nectar guides than those flowers that are predominantly pollinated by the birds, the hummingbirds. All right? Do they occur in the same place? Yeah, you have areas that exist in the same area. But we've started to see that the intermediate phenotype doesn't do any good because it doesn't really attract either of the pollinators. So it's either let's get large to attract those insects, or let's go small and attract the birds. All right? Because the nectar guides, the bird's going to see that and it's going to say, oh, even though I like the large flower, it's going to be problematic for me to get to that nectar and pollinate it. All right? The insect's going to see the small nectar guides and say, it's really not enough space for me to land to get that nectar and pollinate the flower, so I'm just going to pass. So you've got these two different selection pressures. Now, I believe in the paper they even talk about that perhaps we are on our way towards speciation, separating and forming these two species. Right now, you can take these flowers, both traits, reproduce them, and you get this intermediate phenotype, which is one of the reasons why this is difficult to show. You need to be able to produce that intermediate phenotype to show that disruptive selection is actually occurring. They can do that, and then with this intermediate phenotype doesn't do as well as either the larger size or the smaller size. All right? So, how do we model selection? We're going to do this in lab. All right, but I want to introduce you to this idea first. All right, so we have the selection coefficient, and the selection coefficient tells us the proportion of individuals, or a proportion of the genotype that gets removed. All right, we can kind of translate that to a term called fitness. So we already kind of defined what fitness was, right? It's the ability to survive and reproduce, get our genes into the next generation. Well, relative fitness is basically the same term. It's the ability of a genotype to obtain representation in the next generation. But now it's going to be compared to other genotypes in our population. So it's not just a single number. What we're going to do is compare our fitness to every other individual, or all of the other traits that are in that population. And what we then do is scale fitness to 1. So the highest fit individuals have a fitness of 1, and all other individuals that have a lower fitness will be less than 1. This now allows us to look at a population, to look at a trait, and predict how well traits are going to do, or which traits are going to be favored and increase in future generations. All right? So our relative fitness pertains to the genotype. All right? And the genotypes manifest themselves as phenotypes. So the point here with fitness is that if you have the same genotype as another individual, then you usually have the same phenotype as well, which means your fitness is going to be the same. But we're going to kind of throw a wrench in that as we move forward. All right. Relative fitness is affected by the selection coefficient. So the larger the values of our selection coefficient, the stronger the selection that we have. And the stronger the selection against that trait means we have a lower fitness overall. So they're very much related. You'll see it in the lab. I have a table that kind of shows that relationship and shows how you can take your selection coefficient, integrate it into the Hardy-Weinberg, and that kind of relates to our fitness. So I do have this where it says C lab for equations and for practice problems. I think that's it. All right, so before we end this presentation, all right, relative fitness, fitness in general, is a very important concept, and we will talk about this throughout the semester. All right? Fitness relates to a genotype. If you have the same genotype, then you have the same fitness. But there's a couple other properties of fitness that we have to know. All right? So we said property of the genotype. So fitness is determined by our genotype. If you have the same genotype, you have the same fitness, except if you have some phenotypic plasticity going on. And we start that on Friday. All right, so same genotype, same phenotype, same fitness. Fitness is also determined by our environment. All right, so each environment has its own selection pressures. When we change those selection pressures, we change the fitnesses. All right? So we have property of the genotype, and we have a property of environment. So we look at today's environment. Individual that has a specific phenotype, we can say that's the best genotype, that's the best phenotype for this environment. But then if they move and go someplace else, they might not be the best anymore. They might not be the best anymore. So these two things kind of emphasize how you can have variation within a species itself. Individuals, they don't all live in the same environment. All right? And our last property is that if we're going to measure fitness, if we're going to examine fitness, you can only measure it over more than one generation. We can't just go out in our population today and measure it. We have to be able to see what happens to our frequencies in that next generation to have any idea or any hope at calculating a fitness. All right? Because again, fitness is how likely you are to gain representation in that next generation. We don't know that until we view the next generation. All right? So there's three properties of fitness. These are hugely important. You're going to need to know it. You're going to be tested on it. You will be tested on it repeatedly. I can almost guarantee you that this will be on the final exam. Three properties of fitness. It's a property of the genotype, meaning same genotype, same fitness. Property of the environment. Same fitnesses. If you change the environment, our fitnesses change, even if the genotypes don't. And lastly, we can only measure fitness by looking at more than one generation. All right? All right. What I'm going to do, I have a quiz over this presentation. I'm going to release it, but I'm not going to make it due until Sunday night at 11:30. All right? So I know if you haven't taken the quiz that's already posted, you can do that tonight, and then I give you Thursday, Friday, and then Saturday to kind of get it done, and hopefully on Sunday you can go ahead and take it. All right? That quiz is going to be longer. It covers the presentation that we just did. So don't think it's going to be 10 minutes. I think it'll probably be 20 or 25 minutes. That'll be your time limit. All right? We're good. If you have any questions, email me.